Hi, my name is Tom Molnar, and I'm going to be talking to you about my poster titled Genetic Control of Eastern Filbert Blight Resistance in F2 Generation Hybrid Hazelnut Populations. And these are crosses of Corylus americana and Corylus avellana. I would like to acknowledge my co-authors, David Hlubik, who is a PhD student in plant biology at Rutgers University, Shawn Mehlenbacher, who is a professor of plant breeding in the Department of Horticulture at Oregon State University, and then John Capik, who is a research technician working with me on the project. Start out with the introduction, tell a little bit about the backstory here, and then we'll proceed to the materials and methods, the results. We'll touch on the discussion and conclusion, and then of course end with the acknowledgments. As many of you know, the Rutgers Center for Turfgrass Science has been supporting nut tree research at Rutgers University since the project's inception by Dr. C. Reed Funk in 1996. I had the really great fortune of meeting Dr. Funk when I was a freshman in college, only 18 years old, and I essentially helped him build the nut tree program, which I have continued to this day. At first we started with a lot of different nut species: black walnuts, Persian walnuts, pecans, even almonds, pistachios. But since 2006, we realized that hazelnuts were the most likely candidate to have some success here in New Jersey or the Mid-Atlantic region in the eastern United States. So if we look at hazelnuts, the European species, Corylus avellana, is typically grown in regions with Mediterranean-like climates. Unfortunately, most of these European cultivars are highly susceptible to a disease called Eastern filbert blight. And this disease essentially limited the ability to grow hazelnuts in the eastern United States. It's a native disease that co-evolved with our wild American hazelnut, Corylus americana. Now, if we consider the European hazelnut, they're also somewhat cold sensitive. This limits their production to regions with moderated climates. One really great climate is the Willamette Valley of Oregon, which many people know from the turfgrass industry of where seeds are produced. But they can also be grown in the Mid-Atlantic and Great Lakes fruit belt regions of the eastern US and southern Canada. But if we're looking to expand into, for example, northern New York, into New England, or into the Midwest or upper Midwest, the European species isn't really cold hardy enough. Fortunately, you can hybridize the American cold hardy hazelnut, which is also happens to be disease tolerant or disease resistant, with the European hazelnut. The F1 generation of those species, we have found, and others have found, that you can recover disease resistance as well as cold tolerance. But unfortunately, the nut quality, generally because the American parent has such tiny nuts, or poor quality nuts, not very close to commercial quality, the F1 is not good enough to be, for example, released as a commercial cultivar. And what we found was, well, the logical step would be to back cross it to the European hazelnut and recover better nut traits. But in doing so, we realized that the Eastern filbert blight resistance in the American hazelnut was multigenic, and in fact, using a typical or a modified back cross breeding program, we would lose that resistance from the American hazelnut as we move towards a more European background. So that approach wasn't working. An alternative approach is to take those select F1 hybrids between the two species and then create what we would call an F2 generation. And within those progenies, we would expect to see segregation of traits for not only disease resistance and tolerance, but also improved nut traits and hopefully cold tolerance. This has worked in other crops, but it's never really been tried in hazelnuts. And this study, or this poster, represents one of the first attempts at this. So we actually look at progeny developed in the year 2014 and 2015, and now look at them in the F2 generation for their Eastern filbert blight response. in an attempt to try to understand how resistance is controlled or inherited from the American hazelnut. So now I'd like to talk a little bit about the materials and methods. The F1 plants used in this study were also developed at Oregon State University. They were developed from crossing select accessions of Corylus americana, grown and selected by Shawn Mehlenbacher and his team at Oregon State, with eastern filbert blight susceptible European hazelnuts, but European hazelnuts with improved production traits. So a lot of work went into developing these F1s. Big populations were grown out and select individuals were chosen. In this study, 29 of those F1s, representing different families or different unrelated Corylus americana families, were crossed to create F2 generation seed. Now, I should note that those F1 plants were selected in the absence of high eastern filbert blight pressure, so we really didn't know how disease resistant they were. Nonetheless, the seed from these crosses were sent to Rutgers to grow in our field under high disease pressure. And those 29 parents were also clonally propagated and sent as well. So we ended up, from the crosses made in 2014 and 2015, growing out in the field nearly 1,300 seedlings and replications of the 29 parents, 4 trees of each. They were exposed to high disease pressure, and after 5 or 6 years, depending on the date they were planted, they were then evaluated for eastern filbert blight. They were evaluated using a scale of 0 through 5, where 0 equals no disease and 5 equals highly susceptible. Trees rating 0, 1, and 2 would be considered resistant to highly tolerant. Those rating 3 would be considered only tolerant, and those rated 4 or 5 would be susceptible. After that 5 years, we collected all the data. We looked at and calculated progeny and parent tree means for eastern filbert blight response using that 0 through 5 scale. We developed a frequency distribution curve of the entire population of plants as well as individual progenies, and then we were able to take the offspring means and then regress them on the mid-parent values, which would allow us to provide a preliminary estimate of narrow sense heritability. And in fact, this will highlight if there's an additive genetic component of resistance. So now we're going to talk a little bit about the results. We're going to go through each of these tables, but sort of as the main premise here, the results showed that most of the 29 F1 parent trees actually express a significant amount of eastern filbert blight. So if we look at figure 3 here, we can see that really only 3 plants, so 3 of the mother plants, or excuse me, 3 of the parent plants used in this study were actually classified as resistant or highly tolerant. A majority of them rated over 3.8. So 3.8 is getting towards a very susceptible. If you rate a 4 or a 5, you're basically dying from eastern filbert blight. So while these plants were chosen for the study because they had really excellent nut traits, we found when we brought them to New Jersey, they actually succumbed to the disease. But what's really interesting, we did find that many of the offspring, in fact about 1/3 of the total offspring, were actually resistant or highly tolerant. So if we look at figure 4 here, and this is just a histogram that looks at the total number of trees across the population, which is over 1,200, and you can see that 15% were actually rated 0. If we compare that to the 29 parents, only 1 out of 29 parents rated 0. And like I said, most of them rated above a 3, falling into the 4 or 5 category. But here we have a significant number that are 0s, 1s, and 2s. If we look at figure number 5, it shows it in just a little bit more clear way. You can see the offset of the pie chart, where there's 15% zeros, 6% ones, and 11% twos. And from the breeding perspective, even a 3, which we would consider tolerant in terms of a usefulness of resistance, is not terrible. So by taking those parents and crossing them, in fact, we did recover a significant amount of resistance. 6, this shows the proportion of Eastern filbert blight response of those different 0 through 5 classes per each progeny. So that code, the OSU 14068, for example, all the way across there on the x-axis of this chart represents each progeny. And there's there's 6 different colors. Those with the most resistance are arranged on the left, and then moving towards those with the least resistance and most susceptibility on the right. And really what this shows is we did see somewhat of a bell-shaped curve when we looked at sort of the general perspective across the progenies, which we would expect from a multigenic trait, such as what we're finding Eastern filbert blight resistance from American hazelnut. But interestingly, what we did find here also is some progenies were skewed towards having many resistant plants. So if we look to the far left of this figure, you can see essentially no 4s or 5s rated. So every plant in the progeny was either resistant or highly tolerant to tolerant. So we did find a number of progenies that sort of fell that way, where those in the middle of this table, you would find a majority of the offspring were rated around a 3, and that's more of what we would expect, where you're sort of intermediate between the parents. And then some of those offspring succumbed pretty heavily to Eastern filbert blight, and the resistance didn't show through, and that's what we would find on the right. Here we have the regression of the progeny Eastern filbert blight mean on the mid-parent score. And what's important here is the slope of the line, which is 0.638, or we'll say 0.64, and this represents the narrow sense heritability. 0.64 is actually showing a relatively highly heritable trait. And being the narrow sense heritability, this is showing additive gene effects, meaning that if we select F1 parents with a better Eastern filbert blight score, we would expect the offspring mean to be better. So this is moving in a positive direction, and this is really what you want to see from the plant breeding perspective, that we can use phenotypic selection, pick better parents, and then expect to see improvements in the next generation. So this was really exciting to show that we can, even with F1 parents that were not resistant, but they did carry alleles for resistance or tolerance, that we could recover some of those traits in the next generation. Study, we hypothesized that if we grew out large enough populations, we would recover resistance and tolerance, and there would be enough of those individuals to select upon for nut traits. And we were happy to show that that was the case. Nearly 1/3 of the offspring, despite being from susceptible parents, were found to be resistant. And we did see a lot of transgressive segregation, for example, where the offspring were much better than either parent. Now, that with the high narrow sense heritability estimates really reinforces that we're seeing additive gene effects, and that will likely make contributions to this approach in the future. The study demonstrates the presence of additive gene action supporting Eastern filbert blight resistance in the F2 populations. And the fact that we generated a relatively large pool of plants to select from for other traits gives us confidence that this approach of developing F2 hybrid populations will serve us in the future. Of course, this study is the result of many decades of hazelnut research at Oregon State University and at Rutgers University. Funding for this study in particular was provided by the Rutgers Center for Turfgrass Science, the New Jersey Agricultural Experiment Station, the Oregon Agricultural Experiment Station, Hatch Act funds, and multiple USDA Department of Agricultural competitive grant funds. We would also like to acknowledge the help for this study from Emil Milan, Dan Jacobs, Adam D'Angelo, and Dave Smith. Thank you for following along with the discussion of our poster today.